My name is Jun Hee Lee, and I'm from Department of Physiology, Molecular and Integrative Physiology at the University of Michigan. And we work at SenNet as a technology development. So I am directing technology development and application. And we work on our new technology named seq-scope or seq-scope. My background is actually molecular biology and genetics, and my research interest is on the relationship between stress and aging. And we wanted to characterize the cellular and tissue stress responses that can actually work to prevent aging. So that was our major interest. And during the research, we realized that we probably need a systematic tool which can unbiasedly examine the tissue homeostasis and stress response. And that's one of the reasons of how we worked on technology development which can enable doing that.
What drew you to the field of senescence research?
Many people are concerned with aging, and so did I. So when I did my first PhD thesis, I was very naturally interested in aging. But actually, it happened that my experiment didn't work very well at the time. But yeah, so I instead focused on other signaling pathways controlling cell growth and oncogenesis at the time. But when I moved to my postdoc, actually, I was able to find that one of the stress responsive proteins that I focused on, which are named the sestrin family proteins, they were really essentially involved for many different age-associated pathologies. So that's how I finally was able to do some aging research. So I continued to do that. And yeah, that's how I was interested in the field of aging and senescence.
Please describe your work with seq-scope and FICTURE technology. What needs do these technologies address?
So let me start with the sequence scope that we initially developed. So basically, this is the area of spatial transcriptomics. So we wanted to understand how cells and tissues respond to stresses, and we realized that actually cellular stress responses are extremely heterogeneous. So we initially performed some single cell analysis, but we again realized that those single cell analysis destroys the spatial information, and we wanted to get spatial information intact. So we wanted to do spatial transcriptomics, but at the time, the problem was that the spatial transcriptomics resolution was very poor. So it was conventionally 100 micrometer, which is worse than your naked eye, which is approximately around 40 micrometer resolution. So you can see your hair through your naked eye, but former spatial transcriptomics technologies cannot see it because its resolution was too poor. So what we have done is we were able to repurpose Illumina sequencing machine to generate spatial transcriptomics array, which is the development of seq-scope or seq-scope. So it was the first spatial transcriptomics that can provide the microscopic resolution, which can provide some micrometer resolution. And after our development, actually, there has been a lot of other technologies, including sequencing-based and imaging-based technologies that can provide a really microscopic level. But it created a problem in analysis. So when the spatial resolution was low, then each of the spot or spatial unit had a lot of transcripts. And then we are able to do some kind of computational or statistical inference of different cell types and transcriptome phenotypes so that we can understand the data. But when we have this type of super high resolution data, so now our technique can go down to 0.5 micrometer. And if you use some of the imaging-based technology, then you can even get into the 0.2 or 0.3 micron. And then what happens is that in a spatial area, you often has more than millions and sometimes tens or hundreds of millions of different spatial units, which are pixels. But each of the pixels in sequencing based, our technology, it can have a maximum of 10 to 20 transcripts per spatial unit. And for imaging based, it only has one unit. So actually this has kind of made a very serious problem in computation and statistics. So even though we have such high resolution data, we have to kind of collapse the information and actually downsample the spatial information to get the proper spatial inference. So that has been a limitation. So even though we have generated many spatial data sets, we realize that actually the analysis is not capturing all the information that is encoded in that high resolution data. So that's the reason why we kind of wanted to develop some new way of analyzing the data that can show the high resolution spatial data encoded in the data set.
How do seq-scope and FICTURE contribute to your work with SenNet?
Compared to the previous technologies, previous computational algorithms, the FICTURE is really strong because first it can do kind of pixel level inference so that it can exactly point out where all different those spatial cell types are present. It doesn't require cell segmentation, which is extremely challenging for actual tissues. So ideally, if you can have a true cell segmentation, then you can have more precise inference of cell types. But in practice, because in senescent tissue, in inflamed tissue is really messy with all different cell types. So it's very difficult to get a precise cell segmentation. But this picture is able to kind of discern the cell types and then actually make a projections into the histological space without the need of those complicated cell segmentation. So this is really beneficial, especially for SenNet. I think this would be very beneficial because, let's say we identify a senescent cell vector and we wanted to project them into the spatial space. If you do kind of cell segmentation, you may easily lose some, specific senescent cell types which are not usual. But in FICTURE, we are able to kind of project any types of vectors which can be learned from either single cell analysis or analysis of that our own data set, and you can make a vector and you can project them into any types of high resolution data sets. So we have applied FICTURE. So FICTURE was originally developed to kind of understand the seq-scope data. That's why we have developed this FICTURE. But after developing this, we thought that probably it might be possible that this algorithm may work with other data sets as well. So we applied it to other data sets like sequencing-based stereo-seq or imaging-based, Morphish or 10x Genium or nanostring Cosmex, and in all of those applications, actually, FICTURE worked greatly. So basically, you see that in TMCs of SenNet, we are using a variety of technologies, especially high resolution technologies. You can see kind of all different kinds, like, Morse and Genium, nanostring Cosmex, and VGM HD, and stereo-seq, and also seq-scope, and all those data would require their own software to do cell segmentation or kind of specific analysis. But with FICTURE, we are able to kind of analyze them all in the same platform. So it is especially critical for benchmarking and also comparing the different senescence in different data sets. So I think this will open a lot of new possibilities. So that's how we are so excited about that.
What are the next steps for FICTURE?
So, we have developed this seq-scope, and then we have this analytical tool for, named FICTURE. And also we have optimized this seq-scope so that it can have a wider imaging area using more recent Illumina sequencer machine, NovaSeq. And then those protocol papers will be also coming out very soon. And our kind of next vision is that, we can kind of using the powerfulness of FICTURE-based analysis and also using our generated seq-scope data sets as well as the other data sets produced by other technologies. And we thought that probably we may be able to make a web-based interface where we can explore those ultra high resolution data in an interface like Google Maps, so that let's say we have a satellite image like H&E, hematoxylin and eosin, or fluorescent images. So we have those images as a kind of a satellite map. And then we have a gene expression map, we have a cell type inference map, pathway map. So probably we are able to deliver some kind of nice package in web-based interface. And then I think probably looking forward, the data delivery might be more kind of, accessible to general user. I know, I'm a molecular biologist and geneticist, so I know how big the hurdle is for us general biologists to explore any single cell or spatial data set. And probably we may be able to make it more intuitive and easy to use so that many different investigators can get biological insights. So probably that's probably where we are heading to. And we are excited, of course, about also further improving the seq-scope technology to get higher resolution or higher throughput so that it can also give more kind of data regarding spatial and transcriptomic information.
What are your thoughts on SenNet as a community and resource?
SenNet is a really great resource, and we actually got a lot of different ideas and made projects by discussing with the different components. And we have been very active in discussing and arranging collaborations with other TMCs and TDAs, which I really appreciate. And the collaborative environment, I think, which I really like. So and also we think more about kind of different tissue types and different context of senescence so that we can optimize kind of our development into those needs as well. So I think this is a really wonderful consortium.
What would you tell students and junior investigators who are eager to contribute to this research?
I think now it's a really exciting time. New technologies are developed. And also, if you are in this consortium, actually you are in a very nice position to work in, consortium level research. So I strongly encourage you to be actively involved in all these activities. And also, I encourage my students and postdocs as well to do that. And it's really good to learn from different groups and how people do science in this field. So I strongly encourage that for all different SenNet activities you actively participate in, present to what you have been doing, and then also learn from others about recent advancements.